Right. So I will talk about really briefly about how to design a genome sequencing project. So for this, I will go over three major considerations. So first is how do you select your species and the individual you want to sequence, and the next one is choosing the sequencing platforms. And after that, how to get DNA and sequence it. This will be under the context of nanopore sequencing. And finally, I'll give you a quick example of how we did. Um, right, so first, choose your species individual. So not all plants are equally sequenceable. At least for now, there's a huge difference in terms of the genome size. So the smallest genomes we have is this carnivorous plants. It's only 60 megabases. But then you have Paris japonica, this flowering plants that have almost 150 gigabases of genome size, right? So obviously, if you want to sequence, pick a species to sequence, I would go for this instead of the Paris, unless you're interested in a gigantic genome. Um, Also, um, plants have very different plotting levels. And again, if you want to choose, I would choose to sequence this species rather than this ferns that has thousands of chromosomes. Um, Plotting level is very important. Um, right. So the main thing I want to emphasize here is to know your genome size and ploidy level between you start your sequencing projects. And if you want to know the genome size, the Q-Garden C-Value database is very good. It has a lot of genome size informations. So here's an example. I just type in Paris into the database, and it will list their genome size records. So from 1C, 1C means the haplogenome size, and they use the picogram as a unit. So here's a conversion. So you can use 1 picogram roughly equals 1 gig, in a rough sense. Okay, so Paris japonica, huge genome size, but you have something that is much, much smaller than that, right? And this gives you really important information. If you want to generate a reference genome from Paris, you might want to pick tetraphylla or something instead of japonica. Okay. If there's nothing known in your genome in the Q-Garden database or any published literatures you can find, you can consider doing flow cytometry to estimate genome size. The way flow cytometry works is you first isolate nuclei and you stain the nuclei with propylium iodide, and which binds to the DNA and fluoresce. So the more DNA you have in the nuclei, the larger intensity, the higher the intensity that it will give. So basically you have your nuclei, you pass it through this narrow channels, I have a laser coming through these directions, and then you got a plot that look like this. So it's important when you do flow cytometry to include the standards. So in this case, we actually chop, this is a fern that we're interested in. We chop this fern together with tobacco. We know the tobacco genome size, and we chop them together and then um, do this together. We know the tobacco genome size. We know our fern peak around um, three times of the tobaccos. So we know that the genome size is roughly 3 times of the tobacco. Okay. Another way to estimate genome size is from the sequencing data by generating a chimer frequency plot. So this is a series of chimers coming from this sequencing reads. Okay, and the way chimer frequency plot works is you count how many times each chimer appear in your sequencing pools, in your read pools. If it appears exactly 20 times, you will place it here, right? And here is showing that most of the chimers appear roughly 20 times, and that kind of implies that the coverage is around 20x. Okay, so the coverage is a measurement of sequence volumes, how much data you generated, divided by your genome size. So if you know the coverage, of course you know how much data you generated, you can infer the genome size based on the sequencing results. This is using the Illumina data. I will talk more about this later. And there's several handy online tools can help you to do this. So one we use pretty frequently is a genome scope. You can follow the instructions on Genome Scope website, how to generate the data it needs. Basically, you just upload a histogram file, and it will just generate the plot for you and estimate genome size for you. Okay, so that's about genome size. How about ploidy level? A good database we used is the CCDB database. It compiles almost all the known chromosome counts in plants. It's a very extensive database. Again, here, I searched Paris just because that's example I've been using. Paris japonica apparently is a tetraploid, has 20 chromosomes instead of 5. So again, you can avoid accidentally picking up a polyploid just using the CCDB database. And of course, not all the plant species are in the database. There are some handy protocols. For example, this paper that was published recently kind of guide you how to do meiotic chromosome squash. So yeah, again, know your genome size and ploidy before you start your sequencing project. If you work on bryophytes, then you're lucky because they always have ploids. So a reason to work on bryophytes. Right, so now assuming you pick a nice diploid small genome species in your group, then the question becomes which individual you want to sequence, right? There'll be thousands or millions of different plants. Which one do you pick to do your genome sequencing? So an ideal individual should give you a lot of materials. The best, if you can grow them in a greenhouse, or you can turn them into tissue cultures. And this way they can give you a lot of materials to do high molecular weight DNA extraction. This kind of methods tend to require a lot of input tissues. And also allow you to sacrifice different plant parts or developmental stages, or allow you to do some treatments for RNA-seq. RNA-seq will be important for genome annotations and to infer the biology of these organisms, right? And for Hi-C, you also tend to need fresh tissues, right? I will talk about Hi-C more later. All right, so that's, you probably need a lot of plant fresh materials, and it will be best if you have a low heterozygosity, and that will reduce the bioinformatics nightmares and also the sequencing depth you will need. If you can self the plants, that would be great, but if you cannot, just, I will talk about this later. Right, another key consideration is to pick a really clean plant. You don't want to introduce contaminations during a DNA prep. So cleanliness is an important consideration. Right. For genome sequencing, heterozygosity is always not good. Whenever you have heterozygosity, most assemblers will break the context into 2 alleles. So here is you can see this is single context, but then here's a heterozygous site. It doesn't know which alleles to connect to this part. So we break it down into 1, 2, 3 contexts. So if you have high heterozygosity, you will get really fragmented, but also highly redundant genome assemblies, right? And this is not very good unless you want to have a heterozygous genomes. If that's a purpose, then that's a different story. You can kind of gauge the level of heterozygosity by doing Kramer plot as well. So you can imagine if you have heterozygous sites, the heterozygous site will receive half of the coverage than the homozygous sites. So this is a classic heterozygous Kramer plot. This is from a beach that Susie and we sequenced. So you can see this is a main peak, right? And you have a secondary peak right here, exactly roughly the half coverage. And this is a classic heterozygous peak. Okay, so it's always good to shallowly sequence some of the individuals to see what's the level of heterozygosity they have. Okay, about cleanliness. So if you're interested in homeworts like we do, you find a big patch of homeworts and you peel this off, get the DNA and sequence it, you will get a really horrible results. You will have nematodes, you have algae, fungi, bacteria, other mosses. So there's no way you can get a good results by just picking these plants out from the field. So it's important to, in our case, to establish sterile cultures in the lab, that we sterilize the plants, we propagate them exonically, so we have really clean and large amounts of plants to deal with. Right. So again, genome surveys is important. Roughly 20 to 50x coverage will give you quite a lot of information. Will give you genome size, the level of heterozygosity, the repeat content, kind of, from camera plots. We'll also give you some level of contaminations. There's some good programs they can use to assess how many other non-plants reads are present in your sequencing results, right? And this will be useful for polishing your genomes if you're using high long read sequencing. And also you can use this Illumina data for hardware assembly as well. So they will not, to be waste, but it's the information you can get out are very important. Any questions so far? All right. So then the next I want to talk about is the sequencing platforms. There are many different sequencing platforms out there these days. How to choose among them is kind of quite important. So we all know Illumina sequencing, right? It's the most common sequencing platforms out there. It generates a lot of data, but each read is short, usually less than 150 base pairs. So this is not good for assembly, because if you have a repeat longer than the read length, then there's no way you can assemble paths for that regions. And plants tend to have a lot of repeats. So Illumina only assembly tend to give highly fragmented genomes assembly with tens of thousands of different contexts in the end. Right. So you can think about genome assembly is solving a assembly puzzles, a jigsaw puzzles. So you have fewer reads, you have very small pieces. If you have longer reads, you have a larger pieces. And of course, it's much easier to solve the puzzle with larger pieces, right, compared to the smaller ones. And this would be the analogy of Illumina sequencing, and this would be the long read sequencing. And so long read sequencing gives you larger reads, easier to put your genomes back together. Okay, so long read sequencing, single molecule sequencing, similar things. Two companies that offer this, PacBio and Oxford Nanopores, they're based on different technologies, but they all tend to give you really long reads of more than 10 KB and sometimes in the megabase range. The flip side is they have high error rates, so usually 10% of the bases are called wrong. Of course, there are ways to overcome this, but most of the algorithm nowadays can really take those errors into account, and you can either be correct out before the assemblies or post assembly. So the error rate seems high, but it's not a huge issue anymore. And again, there are ways to reduce the error rates in sequencing. So I will focus more on Oxford Nanopore because I think it's becoming very accessible and powerful. It's also more economical, in our opinion, compared to PacBio. So this is the Oxford Nanopore lineup. You have the Mid9. This is the USB sequencer, the most common ones. And Promethean is the most beefier platforms that generate a lot of data. And I heard they have iPhone versions that'll be really fun to work with. Right. So Mid9, this is the device. You plug this into your USB or computer. This is the flow cell. Flow cell is a disposable part. You slot in your flow cells into this device, and you load your libraries into the flow cells, and it will start to the sequencing reactions will start. Okay. So right, more information about Mid9. The most common flow cells now is R9. There's R10 flow cells, but I think R9 is still the most common one. Each flow cells, if you buy them in bulk, costs you roughly around $500. If you use this correctly, or if you are lucky, you got a really good flow. If you are not unlucky and got bad flow cells, you can get roughly 10 to 20 gigabases per flow cell. There's also fungal flow cells, a smaller one. It costs $90, generate fewer data. All right. The library prep, you need to prep your DNA before you put them onto flow cells, onto MinION. Each library prep costs roughly $150, including all the enzymes you have to buy extra and stuff. Takes an hour to do it. The minimum input is around 1 micrograms. You can use less in our experiences. You can also use RNA, cDNA, amplicon. There are different kits for different type of nucleic acids. If you want to get started, you can buy a starter pack. It's $1,000, come with 2 flow cells, and of course a device and a library prep kit. Okay, so that's nanopore. But I also want to give kind of introduce PacBio, give some background about PacBio as well. The really up and coming or really promising thing coming out from PacBio is the hi-fi reads. I have seen some plant genomes assembled using hi-fi reads, and they are amazing. So, like I said, most of the long-read sequencing have high error rates, and PacBio can kind of get around with that by turning your DNA into a circle and have the polymerase go through it multiple times. So you're sequencing the same molecule multiple times, and then you make a consensus of those. Assuming the errors are random, the consensus would be highly accurate. So they can achieve over 99% accuracies with hi-fi reads. And the hi-fi reads will be 10 to 20 kb in length. So you will have highly accurate and long reads. But you can imagine it's because you have to sequence the same molecule multiple times, they tend to be more expensive. At least all the quotes I have so far, they are much more expensive compared to nanopore. But they give awesome assembly. All the assembly I've seen are really amazing. Right. So how much would it cost to sequence a 1 GB genome now? So Illumina data are cheap, right? The quotes we have been getting is around $11 per G. Usually you need 50x to do the genome survey or to do polishing. And then to nanopore, we calculate around $40 per GB, and you need roughly 50x to get good enough coverage. So 1 GB genome, 2,500. It's not bad. Of course, this doesn't take personnel time into account. And the cost is pretty much scalable linearly. So for 1 GB is $25. 5 GB is $12,000. And yeah, it's getting pretty economical now. If you're willing to do the things yourself, make a library prep yourself, do the sequencing around yourself. Okay, so long-read sequencing will give you a set of really nice contigs. Maybe several hundred kilobases or several megabases in length. What you want to do then is, if you want to do this, then is to scaffold them into scaffolds. So find some informations that connect the contigs in some way or not, or the other, and turn them into a scaffold. One scaffolding approach is Hi-C. Hi-C stands for high throughput chromatin conformation capture. And the basic idea is that your DNA, so your nuclei look like this, right? A circle, a ball, and the chromatins of the DNA from one chromatin tend to group together in their own territory, right? So we can capture these conformations that you know the reads coming from that part is coming from the same chromosomes. So DNA from the same chromosome will be spatially close. Okay. So to do high-C, the first step is to cross-link your DNA. So here there are two strands of DNA that are coming from the same chromosomes, but they are distal ends because they're brought together by the chromatin structures, the chromosome territories. Then you cut it like gate, and then you select for the junctions, and you sequence them from both ends. And each two ends were coming from different part of the chromosomes. So will tend to come from different part of chromosomes. And you can take this contact informations and turn your assemblies into maybe chromosomal scale, right? So that's the idea of high-C. Another way to do this is to do optical mapping. So to do this, you take your first you take your high molecular weight DNA, you mix it with restriction enzymes that recognize a specific sequence, introduce a fluorophores. So in the end, you will have a lot of green dots on a string of DNA. Okay. Oh, sorry, my something's not right. Hmm. Anyway, my animation is gone. So maybe I can just draw it then. Oh. Okay, let me try this. So you have your DNA and you have restriction enzymes that recognize it and introduce fluorophores, right? And then you stretch it to a linear form, and you still the fluorophores are on top, attached to it. And then you have a really fancy camera on top. You can measure the distance between this, say 1 kb. This is 500 base pair, and so on, right? So you have a map of where the restriction enzyme are, restriction cut sites are, and you can align this, right, based on the distance between the cut site, and you will get a long stretch of genome map. And you can do in silico cut of your assembly, and then map your assemblies back to this genome map. So if this is your assembly, it has a same cut site here. This is another assembly, another context that have same cut sites, and so on. So you know, counting A, B, C, orient them by this order, and you can then try to fill gaps in between, right? Okay, so that's the idea of optical mapping. Yeah. Okay. So how much would it cost to scaffold a 1 GB genome? For high-C, you can buy library prep kits from, for example, Face Genomics. Each prep costs $500 or so. And then you need to sequence it on top of it. So it comes down to $1,000 for the cost. Again, this is you doing the library prep. And optical mapping is more expensive because I don't think you can operate. That depends on a really fancy equipment. It's called Nano Sci-Fi. So you need to outsource this to outside companies to do this. And the quotes I got is around $3,000 from DNA extractions to running the data analysis. All right. Any questions so far? Okay. The next one is how to get DNA and sequencing on nanopore. Again, this is the do it yourself scenario. You want really good DNA to sequence because the output of your of nanopore largely depends on the integrity of your DNA. Typically, people can either use nuclei prep, so you isolate nuclei and you digest the nuclei, and do things very gently, and you can get pretty good DNA. There are commercial kits, for example, Mac Attracts Nanobind, um, that can help you with this process. We tend to just use what I call the slow CTAB. We pick young leaf, which is be very careful in every steps, no voltaxing. We use cut tips to do all the pipetting to avoid sharing the DNA. This has been working pretty well for us, and it doesn't require us to buy those fancy kits. But again, every plant is different, so you really need to try different methods to see what works for you the best. One way to tell if your DNA is good is to quantify your DNA, and we use qubit. Never trust nanodrop concentrations. Now, never trust nanodrop for quantifications. They tend to overestimate the number of DNA. What you should aiming for is around one micrograms of DNA for nanopore run. And it would be great if you get more, so we can do site selections to get rid of the shorter fragments. The purity is important. You can use purity, or you can use nanodrop to quantify the purity. Um, to get to know the size of your DNA, the integrity part, you can run pulse field gel electrophoresis. This requires special equipment, or you can use bioanalyzers or femtopulse. So Cornell has a femtopulse equipment. So basically, you will give them your DNA, you submit the order, and they will do it, and they will give you results back. But that takes a few days for them to do all the stuff. Um, so we tend to just do low percentage agarose gel and run it for an hour or 2 hours. And we couple that with the NEB 1 KB extend DNA ladder. So the highest band is almost 50 KB. And that this should give us some relative idea about the size distributions of your DNA extractions. And this has generally worked well for us. It's quick. You don't have to walk to a biotech center. You don't have to wait for several days for them to do an analysis. Right, so this is an example. This is from a FemtoPulse QC results. This is a project that Susie and us have been working on together, Canitum. This is a slow CTAP DNA extractions. And you can see the DNA is really good. The peak is around 127 kb. So this is a really high molecular DNA just by real slow CTAP. Okay, but you can see there are some smaller fragments here, and this will impact the sequencing run because they will tend to pick up those shorter fragments and sequence them first. So you want to remove them. And this brings me to size selections. Blue Pippin is equipment that is basically a really fancy and accurate gel extractions machines. You run your DNA for the chips, and then when you hit a certain size, you cut it out and isolate it. It's more expensive because, of course, you need to have this equipment. I think Cornell has this as well, but again, it takes several days, sometimes several weeks for them to get this going, and you tend to lose a lot of DNA in this process. What we end up doing in our lab is to do circulomics, short eliminator kits. It comes with 3 different sizes. Last time I checked, the XS, regular, and XL. Basically, different cutoff for getting rid of the fragments. It's very easy to use. Basically, you mix the solutions with your DNA, centrifuge it, take out the supernatants, and dissolve the pellets, and there you go. That's it. You'll lose some DNA, of course, but in our experience, it's not as bad as the blue pippins, and super fast. The whole process takes like 30 minutes or so to do it. Okay. All right. Nanopore offers several library prep kits for genomic DNA. One is the ligation sequencing kit, rapid sequencing kit, and field sequencing kit. Like I think ligation sequencing kit gives you the best yields and the best results. It takes longer time to do it, but the results tend to be better. So unless you are really hurry, I would recommend doing the ligation sequencing kit. All right, so that's from getting DNA to the libraries, and you can load them on Nanopore V9. Any questions so far? All right, okay. I'll give you a quick example. So we, where is it? All my fancy animation is gone. Gosh, I have to draw it. I have to draw a lamp plant phylogeny. Okay, I can do it. All right, so
your computer is testing you.
maybe I'm in my qualifying exam. I need to draw a lamp plant phylogeny. So we have ferns here, right? The most derived lineage of lamp plants. And sister to it is a clade called sea plants that nobody cares about. The basal to ferns. And then sister to it is lycophytes. And then we have bryophytes, so mosses, leavewards, and homewards. Right. So my group is mostly focused on this lineage. We don't do this. Okay. So we have been sequencing fern genomes. Oh gosh. So we have fern genomes from Salvinia and Azolla. These are the two first fern genomes we did. Then we are trying to branch out to its sister family, Massiliaceae, and we pick Massilia to sequence. Right. Massilia vacita has been a model system to study spermatogenesis, and it's a huge research that's done on this aspect. And it was also the first time RNA was actually used in ferns. So you can actually do some genetics with Massilia. So it's a cool species to work with. We generated nanopore sequencing results or general nanopore sequencing reads using 3 flow cells. And you can see this is a length distributions of the results. The real N50 is pretty decent. We got roughly 33x of coverage. The genome size is around 1 gigabase, and we got 30x coverage. 30x coverage isn't enough to do nanopore only assembly. So we end up using a hybrid assembly. We combine our Illumina data together with nanopore and get and use Masuka. I think you guys will be talking about that later, right? The hybrid assembler. Right. So we got pretty good results. The Contig N50 is around 3 megabases. So this means that half of your genome assemblies are contained in contigs that are longer than 3 megabases. Right. So then we add on top of that high-C scaffolding. This is older data, but anyway, the key thing is after we do the high-C, we can get the chromosome level scale assembly. And then after gap filling, we can really increase our Contig N50 from 3 to over 10 megabases, right? And the whole thing costs us not too much. Defend, yeah, whatever. So less than $5,000. Not including the personnel time. So nanopore sequencing, 3 flow cells plus library prep, Illumina sequencing, and the Hi-C libraries plus Illumina sequencing. Right, so that's a real life example. And actually the end of my presentations. So if you have any questions?
Thank you, Fay-Wei. Nice presentation.
I have a question.
Yes.
How difficult would it be to make like chromosome level type of kind of sequencing, like that you have instead of scaffolds like chromosome level quality?
Right. So you want to have some pretty good assembly to begin with. I think that contact N50 you want to shoot at least one megabases, and then you generate Hi-C data. To generate Hi-C data, you can buy a kit from Face Genomics or Dovetail Genomics, and it's a very long process. If you do it yourself, it takes I think 2 days or something to make the libraries, and you send a library for sequencing. Regularly, Illumina sequencing would do, and then you will map the Hi-C reads back to your assembly, and there are several different pipeline algorithms that can help you scaffold your genomes with the Hi-C data. There are a lot of. Are you going to cover Hi-C, Susie, Adrian, Jacob?
No, we don't. Well, I don't know if Jacob does. I don't.
I do not. Nope.
But if that's something that people have questions about, we can certainly discuss it on one of the breaks or at the very end.
Right. Sometimes they work perfectly. Sometimes you need a lot of manual. The key is it will give you a contact heat map, like basically tell you the contact frequencies based on the read map, the pairs. You can manually move blocks around to make it it can be difficult, but it can be easy. Depends on your genomes and the data you generated. So I cannot say it's easy or difficult because that depends on the organisms they're working with and the initial assemblies and your Hi-C libraries.
Okay. Thanks.
Yeah, I hope that answered your questions.
I'm trying to put an example of one of these heat map things in the chat. We'll see if that actually works for anyone that's curious what that looks like that hasn't seen one before. I think I
had
can.
a really bad one.
Yeah, I have a really bad one. Well, I mean, it's not the worst, but it's not great. Let's see. Oh, it looks like it's not gonna, maybe I can just share my screen really quick.
Yeah. How do I
Let's do
unshare?
that.
Okay, here we go.
My stupid computer will actually cooperate. That'd be great. Okay. All right, can everyone see that?
Yep.
So as I said, this isn't really the best one, as Fay-Wei can probably attest to, but essentially, whenever you do this, you want to have the largest contigs as possible, as Fay-Wei was saying, and then the more you can get these things sort of arranged into boxes. So we're looking at the Hi-C reads and basically how they're mapping to the assembly. And so it's arranging things that map a little bit closer together and trying to turn those into chromosomes. So these boxes are in theory chromosomes, but you can see like you get some messy things like here. And in plants, these things are always messy because there's always interactions between like telomeres and centromeres and telomeres of another chromosome. So you get all of this noise that's sort of out here. But there's a tool called Juicebox that you can use to manually edit these. So you can try to like clean up things that look like errors. And in my experience, Hi-C almost always produces some odd errors, either putting things together that actually aren't on the same chromosome, or sometimes you'll have inversions, so it flips something in the wrong orientation. So it's always nice to use one of the other methods that Fay-Wei was talking about, like optical mapping, or if you have a genetic map, to like as an independent data point to see how good your high C scaffolding was, because there's usually some problems. Was there anything you wanted to add, Fay-Wei?
Not really.
I'm going to put my
I hope
messaging
there
that
are
way.
more options for high C analysis. Right now, it seems I only have the 3D DNA.
Yeah, 3D DNA has always performed the best when I've done it, but there's still room for improvement. I know
Yeah.
there's some new one that's supposed to work with polyploids, and I haven't tried that yet. I'm failing to remember the name right now, but
We tried all high C. I think that's the one you are referring to.
yeah.
We got weird results. But
Yeah, I tried salsa and what was the other, like the original one? I think it starts with
L,
an L.
yeah.
And I basically just did it by hand and got a better result in Juicebox. So yeah, there's definitely some room for improvement there if anyone wants to develop some bioinformatics tools. Are there any other questions for Fay-Wei?